networks. Next up, we have 3 very exciting contributed talks. And first is Fritz Bayer. Fritz is a PhD student at ETH Zurich, where he works at the intersection of causal machine learning, genomics, and personalized medicine. Before his PhD, he studied physics at the University of Freiburg, Imperial College London, and the University of Oxford. And in this talk, he'll show how personalized treatment of cancer patients could benefit from causal knowledge about our data. Take it away, Fritz.
Okay, well, thank you for this very kind introduction. I think this is actually a fantastic workshop, and I'm very happy to be here. So in my talk, I will talk about network-based clustering of pan-cancer data accounting for clinical covariates. So in other words, I will show you a method that allows to cluster patients based on mutational profiles. And in particular, I will highlight why the clinical covariates play a role here and how we can integrate them in the clustering. So as a motivation, let me start quite general by saying that cancer occurs when cells are struck by mutations in their genome, which makes these cells grow faster than they should, right? And since we can measure these mutations, we are able to define the overall goal of identifying subgroups of patients that share biological properties based on these mutational features. Now, while this is clearly a challenging goal, at the same time, we also need to clarify that every step that we take towards this goal is one step towards the personalized treatment of patients diagnosed with cancer. Right, so why is that such a challenging goal? So this is generally discussed under the main problem, which is called the heterogeneity of cancer. And just to illustrate what we mean by that, consider the following example where you have a patient and that patient can have up to 4 possible mutations in the genes X1 to X4. Right, so how many possible combinations can we have here? So the patient can either acquire 0 of these mutations, marked here in gray, or he can acquire all of these mutations, marked here in blue, and basically any combination in between. So overall, there are 2 to the 4 possible combinations that can be acquired. And if we start including, for example, a clinical covariate such as the biological sex, that space grows even more. And already for that simple example where you have only 4 genes and 1 clinical covariate, we already have 2 to the 5 different combinations that we can consider. Right, so this space is massive, and at the same time, it's not like each and every of these single combinations necessarily represents a biologically distinct behavior of the cell, right? So you might actually see that some mutations are more relevant to the cell's behavior than others, and some mutations, so-called driver mutations, make even other mutations more likely to occur in the first place. So there are probabilistic relationships within these genes, and the same holds true for the clinical covariates. As for example, the biological sex will have an effect only on specific mutations here. Right, so we can actually exploit these probabilistic relationships in the data in order to compactly represent this huge combinatorial space in one probabilistic graphical model. And what we chose here is the directed version of probabilistic graphical models, which is a Bayesian network. So in that Bayesian network now, each edge represents a probabilistic relationship between the variables. So for example, here sex has an impact on the gene X2. Right, so this is essentially the perspective that we want to take. So we propose a network-based clustering method that allows to cluster mutational and clinical covariate data based on their distinct probabilistic relationships. And this is essentially outlined here in this huge plot, where on the left you can see the input data, where we have on the one side the mutational data from patients that acquired different cancer types, marked here by different colors. And we have the clinical covariate data, such as, for example, age and the biological sex. Now, we take this input data, and from that we learn different clusters, where each cluster is now associated with a distinct probabilistic graphical model, so a distinct Bayesian network, right? And in particular, there are 2 things that I'd like to highlight here, and that is, first of all, why, when we are interested in the mutational networks, why we actually need to integrate the clinical covariates and how we can integrate them. So before I dive into that, let me just briefly give you an introduction to Bayesian networks in case you're not familiar with them. So a Bayesian network is constructed of a directed acyclic graph that we can see here on the right, where the edges in between the nodes represent probabilistic relationships. And kind of the core and the heart of a Bayesian network really is this factorization property, which essentially means that the probability of a single node is conditioned on the parent. So as an example, we can see here in this variable x2, the probability of x2 is conditioned on the variable x1, which is the parent of this variable. Right, so understanding this, I can actually highlight now why the clinical covariates are so important when we want to learn the mutational networks. So consider a simple example where you have the biological sex, which has an impact on the genes X1 and X2, right? So for example, these might be two genes where the mutation might be more often observed in male patients. So if we were to exclude these covariates from the analysis, we would first of all lose the accuracy of modeling the mutations because we can't condition on the biological sex. And second of all, we might also introduce a confounding effect, because the fact that sex here confounds X1 and X2 leads to a correlation within these two variables. And if we later then learn the network, that is likely to lead to a false edge in the network. Right. So on the one side, we clearly need the clinical covariates in order to accurately model the mutational data. And on the other side, we actually don't want to cluster or perform a clustering based on the clinical covariates. So why do we not want that? Because if we do so, we, for example, could end up with one cluster that has mainly male patients in that cluster and another cluster where we have mainly female patients. And that is clearly not desirable since we already have that information. And in the end, we can always stratify by the sex. Right. So in order to essentially overcome this obstacle and remove the effect of the covariates on the clustering, we propose the following covariate adjusted membership probability. And without going too much here into the details, there's one thing that I want to point out, and that is that the probability here of the mutations is conditioned on the clinical covariates. So the mutations here are XV and the clinical covariates are XC. And that essentially allows us to model the mutations based on the clinical covariates, but at the same time, we also remove the effect on the clustering in the clinic of the clinical covariates. Right, and we show in our paper that actually this leads to a lower variance in the cluster, so in the clustering process. Right, so at the same time, we were also interested in seeing how that works out in simulations. So what we did is we compared our method against standard clustering methods, where we included an increasing number of clinical covariates or covariates in general. So the methods that we compared to were a Bayesian network mixture model, K-means, and a Bernoulli mixture model. And since these standard clustering methods don't account for clinical covariates, we essentially had to apply them twice. Once we applied them on the mutational data alone, and once we applied them on the mutational and the covariate data by treating both variable types the same. And what we see here on the y-axis is the adjusted RAND index, so higher is better. And we see that with an increasing number of covariates, we significantly outperform these standard clustering methods that do not account for the covariates. Right, so while I think it's interesting to see that this works in theory and in the simulations, that the main question obviously is, how does it look in practice? So in order to evaluate that, we analyzed a large genomic data set of over 8,000 cancer patients diagnosed with 22 different cancer types. And essentially, we used our method to cluster these patients into different groups. And to, in the end, evaluate whether these groups are biologically meaningful, we performed a survival analysis that you can see here on the right in this Kaplan-Meier plot, where you can see the survival probability of the patients associated with the different clusters labeled A to V. And what essentially came out of this analysis is that our clusters are highly predictive in survival. And just to put this into perspective and compare it against the best performing method of our numerical simulation, we're actually able to learn clusters that are significantly more predictive than the standard methods that do not account for clinical covariates. And I think what is particularly interesting to look at is the scenario where we only analyze the mutational data and completely neglect the clinical covariate data, because here we have a significantly worse performance compared to our covariate adjusted method. Okay, so I think what is also interesting here to point out is that we were not only able to learn clusters that are more predictive in survival than the standard clustering algorithms, but we also were predictive. We were significantly more predictive than the clinical information, such as, for example, the cancer type or the stage. And I think this is particularly interesting because it's tied to the hope that our cancer-specific networks can be used as clinical biomarkers that may be able to predict the treatment outcome and hence allow for more personalized treatment of cancer patients. And this is in fact ongoing work, and we have a paper coming out on that soon where we analyze these cancer-specific networks found in acute myeloid leukemia. So with that, let me summarize. I've proposed you a method that is a network-based clustering method that integrates mutational and covariate data in networks of their probabilistic relationships. And we saw in simulations that this outperforms standard clustering methods. And in pan-cancer data, we saw that we could identify clusters which are predictive of survival beyond the clinical information. Right, so with that, I'd like to thank you for your attention. Also, thanks for Jussi Moffa, Nico Berenwinkel, and Jack Kuipers for the contribution in that work. And I'm happy to take questions if you have any.
Wonderful talk, Fritz. Thank you.
Thanks.
So for folks in the audience, this would be the time to enter any questions if you have them into the Q&A, and I can surface them because we have about a couple of minutes or so. But in the meantime, I do have one question for you, Fritz, is that Bayesian networks have this kind of notorious reputation for poor scalability. Is this a problem that you encountered throughout this study?
Yes, in fact, in order to essentially handle that scalability, we only included in the analysis the most significantly mutated genes, right? So instead of just analyzing all mutations that we could have had, we've really focused on the most significantly mutated genes in order to learn these networks. And even in that case, you see that we saw actually that for the simulation, we had a very low runtime of only a couple of minutes. But when we actually analyzed then this huge data set of over 8,000 cancer patients, where we had actually the mutational profiles of where we essentially considered more than 200 genes, we had a runtime of almost 1 day, so 23 hours.
Great, thank you. And we have one question in the Q&A, and I think we just have a minute for that. So a question from Ben: can you discuss any more about which features separated the clusters? And do you know how these differed from other methods naive to confounding issues with clinical covariates?
Let me go to a slide here. So the different clusters are now associated each with a distinct Bayesian network. So the difference really in these clusters is that the probabilistic relationships in the clusters are different. So in the end, we learn clusters where the patients in these clusters share probabilistic relationships, right? And that, in fact, is different, for example, so here also the fact that we integrate here the covariates is different from the standard methods in the way that we here are able to deliver more accurate results and also remove potential confounding effects.
Great, thank you. I think there was one more question in the chat, but we can take that offline in the interest of time since we have to move on.
Sounds great.
Thank you again for the excellent talk, Fritz.
Thank you.